Welcome to Stem Cell Research in Neuroscience. In this video, I will introduce you to this unique field of research by breaking down what it is, how it works, and where it's going. Did you know that 1 in 6 people worldwide live with a neurological disorder? That's almost 1 billion people with 6.8 million deaths each year. From Parkinson's disease to stroke to traumatic brain injuries, neurological disorders encompass a range of conditions that affect people from all demographics. Although there are effective treatments available for many of them, they are largely engineered to reduce symptoms, not necessarily repair damage to the brain. This is where stem cell research can help. Stem cell research with respect to neuroscience is used to inform stem cell therapy for neurodegenerative diseases and brain injuries. An acute characteristic of these conditions is cell death. Therefore, the main aim of stem cell research in neuroscience is to replace dead cells in the brain with healthy ones. But what exactly is a stem cell? A stem cell is an unspecialized cell in the human body that has the unique ability of self-renewal and is able to transform into specialized cells. In this context, unspecialized refers to the lack of tissue structures required to perform specific functions. Therefore, highly unspecialized cells can transform, or in other words, differentiate to a greater extent. Stem cells exist in two forms, adult and embryonic. The differentiation of adult stem cells is limited to cell type or lineage, in phenomena known as unipotency and multipotency respectively. On the other hand, embryonic stem cells are pluripotent, meaning they can transform into any other cell type in the body. Both cell types can be linked to neural stem cells, with embryonic neural stem cells acting as a precursor to their adult counterparts. First, let's take a look at embryonic neural stem cells. In the early stages of pregnancy, the mass of cells that will eventually become the fetus is known as the inner cell mass. During embryonic development of the central nervous system, a small number of neural stem cells from the inner cell mass transform into radial progenitor cells, also known as radial glial cells. These cells reside in the ventricular zone, where they generate large amounts of the three main cell types in the CNS, oligodendrocytes, astrocytes, and neurons, through a process known as neurogenesis. This process eventuates in the formation of the entire brain, which in turn begins to produce adult neural stem cells. Adult neural stem cells are predominantly located in two main areas, the lateral ventricles and the hippocampus, both regions in which neurogenesis has been proven to persist throughout adulthood. In the subventricular zone, formerly the ventricular zone of the lateral ventricles, and the subgranular zone of the dentate gyrus and the hippocampus, two adult neurogenic niches can be found. A similar process to one mentioned earlier occurs, wherein progenitors create neurons that migrate to other parts of the brain. Adult and embryonic stem cells respectively form the basis for endogenous and exogenous approaches to regenerative medicine. The endogenous approach involves mobilizing resident adult neural stem cells and supporting them with growth factors. Take stroke, for example. Research has shown that the brain induces neurogenesis in response to ischemic injury in a three-step process that involves the migration of adult neural stem cells to the site of injury. However, the neurons that are produced have a very low chance of survival due to a lack of trophic molecules in the surrounding environment. During treatment, the patient may be administered with growth factors, such as erythropoietin, epidermal growth factor, and insulin-like growth factor, to stimulate neuron formation and enhance survival in the damaged area. Additionally, the endogenous approach is not limited to stroke, and may also be applied to conditions such as brain tumors. In contrast, the exogenous approach to stem cell therapy relies on extracting embryonic stem cells, cultivating them in vitro, and transplanting them into the brain. Embryonic stem cells isolated from the inner cell mass can be made to differentiate into the neurons required to produce neurotransmitters relevant to the disease and its treatment. For example, in the case of Parkinson's disease, clinical trials have been conducted where an embryonic mesencephalic tissue rich in dopamine has been cultivated and subsequently transplanted into the striatum, where they re-innervated neurotransmission to restore dopamine levels in the patient. Much like the endogenous approach, the exogenous approach can also be applied to other conditions, such as Huntington's disease, dementia, and ALS. While these breakthroughs in neuroscience appear very promising for the field of medicine, it is important to remember that stem cell therapy is not without its setbacks. Firstly, the efficacy of stem cell therapy is still quite moderate, with clinical trials demonstrating little to no improvement after treatment in many cases. Stem cell research has also experienced significant roadblocks in the form of conflict between the desire to make progress and the absolute need to proceed with caution. As is the case with all fields, potential benefits of the research must be weighed against the risks of harm at every stage. In this regard, research has faced heavy criticism pertaining to the extraction of embryonic stem cells. Ethical concerns aligned with the pro-life movement have been raised regarding the belief that life begins at conception and an embryo is consequently a person. Moreover, the behavior and mechanism of stem cells has not been understood completely. There are still many unknowns in the realm of possible side effects and longevity of various treatments. That being said, these limitations provide opportunities for further research in the future. Once researchers understand how stem cells operate and interact to the fullest extent, progress can be made in growing human embryonic stem cells in a laboratory setting. Not only will this bypass the aforementioned ethical barrier, but it will also create opportunities to explore other areas of the nervous system. In particular, the spinal cord can greatly benefit from even partial restoration by stem cells, allowing patients to regain lost functions such as movement. The implications of stem cell research in neuroscience are far and wide, and new advancements are being made every day. Slowly but surely, the capabilities of stem cells are being harnessed to forge a path towards a world where humans can be free from neurological disorders.